Welcome to Human Anatomy and Physiology. This will be the first in a series on the fundamentals of the nervous system. Chapter 11 gives us an introduction to the nervous system, which will provide a foundation for the subsequent chapters, which will take us right down to the end of the semester. You can think of the nervous system as a group of sensory neurons which will transduce or convert energy or stimuli from the outside of the body into impulses, action potentials, which you're familiar with, which enter into the central nervous system, the integration center, which just means putting everything together and making sense out of it. And then some neurons will carry information out. Those are called motor neurons, and they will activate effectors. They will make something happen. It will react to the sensory inputs that come into the nervous system. So that's a pretty simple way of thinking about it. So what we're going to do is, just as we go through various parts of this topic, we're going to take a look at the sensory neurons, see where they're located, what their properties are. We'll look at various parts of the brain and brainstem and spinal cord, and then we'll take a look at some of the motor neurons that control some familiar things like skeletal muscles and some other parts of the body. So here's another way of looking at that, a little more details now. Looking at the central nervous system, the brain, brainstem, and spinal cord, which is the integration center, and then the peripheral nervous system. We have from the peripheral nervous system information coming into the central nervous system. That's sensory information. And we also have information that once decisions are made going out from the CNS to the peripheral nervous system. Well, there's more than one division, afferent division of the sensory components. There are somatic sensory neurons, which are the so-called voluntary ones, the ones that you know about, right? The things that you can feel. Sensory neurons send information into the CNS, but there's also some visceral sensory neurons, or a visceral division, where there's stuff happening inside your body that you don't really know about on a daily basis, right? The control of your digestive tract, for example, or your heart. Those things just happen. Outgoing information is called motor information. The motor divisions carry that information to specific locations. The somatic motor division is the one you're familiar with already. It's the sum of all the neurons that carry information to stimulate skeletal muscle motor units. But there's also an autonomic division. There's an involuntary component there too. There are two parts to the autonomic nervous system. The sympathetic division, you've probably heard of that, the so-called fight or flight division, and here we have little cartoons showing the direction of things is actually going out from the central nervous system to the viscera or organs. The sympathetic nervous system might be cranking up your heart, right, if you get excited or exercising. And the parasympathetic nervous system, the so-called rest and digest system. So that's more of a maintenance part of the autonomic nervous system. So we'll be taking a look at each of these in turn. You can see that there's like this sort of box diagram looking thing. I just wanted to point out that, for example, this box, the peripheral nervous system, encompasses all of these things down here. We're just doing kind of a breakout to show some of the distinctions between parts of it, but it's not separate from the peripheral nervous system. It is the peripheral nervous system in a sense. Okay, let's take a look at the tissues first. We'll get right down to brass tacks. What do we find in nervous tissue? Well, neurons, we've already mentioned back in chapter 4, are the information carrying cells of the nervous system, and they're the ones that get the most press time. But it turns out there's quite a few supporting cell types that are important for the neurons to do what they do in the nervous system, and those are called glial cells or neuroglia. So there's a number of different types of glial cells that are illustrated here. Let's take a quick look at some of the glial cells. First, we'll look at glial cells that reside in the central nervous system. Astrocytes are the most abundant neuroglia, and they're kind of supporting cells. They physically support the neurons and the parts of their processes. Neurons have long thread-like processes, and they wrap up the capillaries in the central nervous system. So these astrocytes seal up the surface and create part of what's called the blood-brain barrier. The capillaries in the central nervous system are extremely tight as it is, And so they're pretty restrictive about what can diffuse out from the blood into the interstitial fluid here. But the astrocytes then add another layer onto that, and now we have what's called the blood-brain barrier. It's a very privileged environment here in the interstitial fluid of the brain, for good reason, as we'll discover in many instances going along our way. Another function of these astrocytes is to not just control what diffuses out of the blood into this fluid, right, controlling the composition of the fluid, but also to clean up excess materials that are going to accumulate out here otherwise, right? Neurotransmitters are coming from axon terminals, just like at neuromuscular junctions, and they tend to diffuse away, and they would accumulate in this fluid and kind of short circuit things, but they don't, because the astrocytes will take up those excess neurotransmitters, and indeed all the potassium ions that are released during repolarization of neurons after action potentials. Okay, microglia are phagocytes of the central nervous system. CNS has its own phagocyte population. The white blood cells that roam throughout other parts of your body are excluded from the central nervous system. It's a privileged environment in there, and normally things are kept pretty clean in there, and there's no need for any. But occasionally you could have an emergency in which there's trauma, damage to the barriers that keep stuff out of the CNS, and an infection may occur. In effect, then there would be inflammation and white blood cells going in there, causing damage as well as healing. All right, ependymal cells. There's some fluid-filled hollow regions within your brain and spinal cord, and they're filled with a fluid called cerebrospinal fluid, CSF. And in fact, the cerebrospinal fluid is completely surrounding your brain and spinal cord. In fact, it's like a bowl of water in which your brain is floating and bobbing, so that just like when you get in the swimming pool, you have this tremendous buoyancy. You can just jump off the tip of one toe. Well, that's how your brain is. It's kind of coddled in there by floating in the cerebrospinal fluid. Where does it come from? Ependymal cells. They're a type of epithelial cells that are ciliated, and they will secrete this ependymal, or this CSF fluid, which they produce from blood plasma, the water of blood plasma, and then they can make it flow. There's actually a current of CSF, and we'll take a look at that a bit later in chapter 12. So they also form part of the blood-brain barrier, though, because if you can imagine a bunch of blood capillaries down here in the vicinity of a lot of ependymal cells, there's fluid filtering out of those blood capillaries, and these cells will then modify that fluid and produce cerebrospinal fluid. But that represents a pathway potentially into the inner sanctum here, the central nervous system, from blood through the ependymal epithelium and into the CSF. Oligodendrocytes, awesome mouthful of a word. These are cells that myelinate axons in the central nervous system. You've heard of myelin sheaths, and you've probably heard of multiple sclerosis. Well, myelin sheaths are wrappings of phospholipid plasma membrane-like layers around axons, the long thread-like projections or processes of neurons. And so these oligodendrocytes wrap up and insulate those things. And in fact, as we'll see in just a little bit, the myelin sheath is important for the speed of communication of neurons. So here's an oligodendrocyte, and it is actually going to myelinate or wrap up a little segment of a bunch of axons. This looks like maybe it's just an artist's conception where we chopped off the myelin there so we didn't have to draw it. It actually does stop right here. There'll be a little gap, and then another oligodendrocyte would myelinate the next little segment of the axons of multiple neurons. This oligodendrocyte might myelinate parts of maybe 50 axons in its vicinity, and then the next oligodendrocyte would take over from there, and then the next, and so forth. So anyway, multiple sclerosis is an autoimmune disease in which the myelin sheaths, which are so important for the speed of conduction and insulating these axons, is being attacked and destroyed. There's ominous looking little X right there. In the peripheral nervous system, there are some glial cells. And there's a different kind of cell that myelinates the axons in the peripheral nervous system, equally as important for speed of conduction, to get information where it needs to go, like right now. And we can give some examples in a little bit of how important this is. So that's what you see, these little buttons of myelin sheath right there along an axon in the peripheral nervous system outside the brain and spinal cord. And then the cell bodies of neurons, right? We'll get to those in a second. They are kind of wrapped up and protected by some so-called satellite cells in the peripheral nervous system. I won't say anything more about them because as far as I know, there's not a really well-known physiological role for satellite cells. So here's a little cartoon illustrating a Schwann cell and how it myelinates its own little segment of the axon. Remember, each Schwann cell myelinates one little part of an axon, right? Not like oligodendrocytes that reach out to a bunch of different axons. This Schwann cell lives right here, and as it goes through the process of development, it starts synthesizing cell membrane, and it just wraps the cytoplasm and nucleus. You can see here in this thicker part, it just starts to wrap around and around this axon, producing layer upon layer upon layer of myelin sheath, and that's how it insulates, produces many, many layers of phospholipid bilayer there. And here's a beautiful electron micrograph of the myelin sheath on a great big axon in the peripheral nervous system. Pretty cool. Incidentally, this bit of the Schwann cell, where its organelles and its nucleus and its cytoplasm reside outside of this sort of spiraling layering of the myelin sheath, is called I'm sorry, it's escaping me, not plasmalemma. It'll come to me in a minute. All right. Here is a prototypical neuron. And we'll highlight a couple of features of neurons, and then we'll get a little more differentiated about things in just a second. But this is like a prototype, right? It's a neuron which has a bunch of antennas or dendrites which receive the incoming information. It's going to receive information maybe from some other neurons, and then it might transmit information. So the dendrites are the receiving part of the cell, and in fact the cell body, called the soma, also can act as a receiving region. So all the yellow parts up here, and then where you see the myelin sheaths, the Schwann cells here, that's the axon. And the axon is where action potentials occur, and they are propagated or spread right down the axon to its terminals. And then, just like on a motor neuron of a skeletal muscle cell, neurotransmitters would be released, which would then bind to receptor channels. So there's a lot of overlap between what we're going to see going on here in the nervous system and what we've already seen with motor neurons of muscle cells and muscle cells themselves. So that's going to be a really great advantage, because we already have begun to understand certain features of how neurons do what they do by way of communication. All right, so these cells have a lot of endoplasmic reticulum, which is some of the organelle structure that's involved in synthesizing a lot of where the neurotransmitter is going to come from, and all of the phospholipids that are going to make up vesicles, right? We're going to have a lot of vesicles filled with neurotransmitter here in the terminals. So all that stuff has to be built in the cell body. And then all the vesicles full of neurotransmitter and so forth are going to be shipped down the axon along a cytoskeletal highway by energy utilizing little transport machines. Coolest thing ever, right? We're going to attach those vesicles onto a little transport machine, which is going to go right down like a train track down the cytoskeleton using ATP to bring those vesicles to the terminals. And then if we have some debris and some leftover membrane bits and some broken down neurotransmitter in vesicles here in the terminals, that can be again attached onto a different molecular motor and shipped right back to the cell body for recycling. Pretty cool stuff. Lots of mitochondria. These things are super metabolically active. Neurons are very metabolically active. You've got to have plenty of oxygen coming in, because you've got to do aerobic respiration, get the 32 ATPs per glucose. You can't be fooling around with anaerobic respiration here in the nervous system at all. There's actually you can see a little microheterogeneity, the nucleolus within the nucleus. I would also mention there are some dark spots, dense spots in neurons that are called Nissl bodies, N-I-S-S-L. Isn't that strange? Nissl bodies that are characteristic of neurons. One of the first things they discovered when they were studying neurons in the microscope, so they've stayed around till this time, and that's the last thing I'll probably ever say about Nissl bodies. Neurons are eternal, essentially. You have your complement of neurons for the most part when your nervous system reaches maturity or your early 20s, and all those neurons are going to stick with you for the rest of your life. minus a few that might get deleted here and there as you age, unfortunately. These cells are amitotic. They have no centrioles. They can't undergo mitosis in most instances. That's what I mean by amitotic. Mitosis is cell division. A means not dividing. So these cells are not going to divide anymore, except in part of your brain. It's been recently discovered that in the hippocampus, in areas where there's memories being formed, long-term memories being formed, learning, you actually create some new neurons during those processes. That's a cool thing too, because the party line for many, many years was no new neurons, read my lips. Okay, so there's a beautiful soma and some dendrites and maybe the axon going out from a neuron in this beautiful EM. Let's do one last thing before we end this first part of our discussion of the nervous system, our introduction, and that is to classify neurons according to type, and we'll maybe even say a little bit about their jobs, what do they do. So let's look first at this neuron. It looks just like the prototype neuron we just looked at. It's a multipolar neuron. That's because it has a whole bunch of dendrites, right? A whole bunch of processes here that are the information gathering components, and the cell body, the soma. Then there's a long thread-like axon carrying information somewhere, and some axon terminals which can release neurotransmitter. As we'll discover in just a little bit, the only place that an action potential occurs on this cell is on the axon. Remember when we talked about the neuromuscular junction, we found out that when acetylcholine binds to its receptor channels in the neuromuscular junction, we produce graded depolarizations. The more acetylcholine, the more open channels, the more the membrane depolarizes. As sodium ions then diffuse to the periphery, to the perimeter, that's when an action potential may start. If we reach threshold and a sodium electrogenic channel opens, then we trigger an action potential which will spread down the entire surface of the muscle cell. Well, it's the same kind of thing here with this neuron. We have graded depolarization that may occur on the dendrites and cell body. Action potentials begin, if they're going to begin, at the start of the axon, and then spread right down to the terminals. Nothing can stop it. Okay, so that's a multipolar neuron. Let's quickly say a little bit about where we may find them. Multipolar neurons are all of the motor neurons. I guess we'll probably see that in the next slide. And all of the interneurons. We'll say more about that. Okay, let's look over here at a unipolar neuron. A unipolar neuron, here's its cell body, its soma, and it only has one process emanating from the soma, so it's called a unipolar neuron. It has some dendrites. Its dendrites are way over here, and it has a long axon and some terminals, but the cell body is not involved in that receptive kind of process. So this is a sensory neuron. So its dendrites are in some region of the body that either from the outside or from internal organs is going to receive some information, transduce a stimulus into impulses, and then send those impulses or action potentials down the length of this axon to its terminals, and the terminals of the sensory neuron will be in the central nervous system. So it'll have its dendrites out either at the surface of the body or in an organ, and it will deliver its information by way of neurotransmitter exocytosis in the spinal cord or the brainstem or the brain. Pretty cool. Whereas this motor neuron, its cell body may very well be located in the central nervous system, the spinal cord, and its axon would carry its impulses to its terminals, perhaps in the periphery. But all the interneurons, what do I mean by interneurons? I've written that in there for you. When sensory information comes into the central nervous system, there are many, many, many unimaginable numbers of neurons that form circuits that process all the information. All those neurons that are doing all that processing are called interneurons, and then we send information out by way of motor neurons. So we have a relatively small number of sensory neurons, or unipolar neurons, a unimaginably large number of multipolar interneurons that form the mass of the brain, for example, in the spinal cord, and then we have some motor neurons, a relatively small number of motor neurons carrying the information out to the periphery. So that's our classification of neurons. Here's like a anatomically correct look at maybe a couple of examples of some multipolar neurons and unipolar neurons. These are like the Barbie drawings, right? They're so perfect. They're so simplified. Here's some actual maybe neuron cells that are drawn as faithfully as possible to how they might look in the actual body. And so here's another way of looking at this. We see interneurons are multipolar neurons, and then motor neurons, going in this case from the spinal cord out to the periphery to activate an effector. In this case, it's an alpha motor neuron of a muscle cell, skeletal muscle cell, I should say, or skeletal muscle. Unipolar neurons are sensory neurons. So here we see in this example, dendrites at the surface of the body, right? If something poked your skin, you'd feel it. Some impulses would be transmitted into the central nervous system, and the terminals of this neuron would be here, and they would synapse or impinge on another sensory neuron here, which would carry information up to your brain. So it's pretty cool. So you've noticed probably by now that I'm ignoring this middle column right here. We'll only talk about bipolar neurons when we talk about special senses, hearing and vision. That's the only time we're ever going to encounter any bipolar neurons. So let's ignore them for now. Just focus on multipolar neurons and unipolar neurons. So when we pick up next time, we'll see that all these neurons have a very negative interior, negatively charged interior, a resting membrane potential, just like a skeletal muscle cell. We'll also discover that in order to get these things to do something fun, we're going to create or generate action potentials in exactly the same way as we did on muscle cells. So we're just going to review that. This is the beauty of this chapter, is it does a really nice job of reviewing what's a resting membrane potential and how does it come about, and especially what's an action potential and how does it propagate or spread along the axon of a neuron. And then we'll get into more details about the nervous system more specifically.